Hello everyone, welcome to Exam Essentials. Today we will solve IGCSE June 25 Paper 2 R paper. So let's jump right into it. So here we have a passage about pollination in fruit trees. And plants need to be pollinated to produce fruits and seeds. Some plants are self-pollinated and others are cross-pollinated. Self-pollination is usually the transfer of pollen from flowers on the same tree. In fruit trees, self-pollination also includes the transfer of pollen from another tree of the same cultivar. A cultivar is a genetically identical group of trees produced by selective breeding. Self-pollinating plants need bees or other insects to transfer pollen. Example of self-pollinated fruit trees are palm, apricot, and peach that have sweet-tasting soft fruits. Other fruit trees require cross-pollination and only produce fruit by fertilization from a different cultivar. Cross-pollination is the transfer of pollen from one cultivar to the flower of a different cultivar. Self-incompatibility prevents the same plant or cultivar from fertilizing its own flowers. For example, the pollen from one cultivar of an apple tree will not fertilize trees of the same cultivar. Most hard fruit trees such as pear and apple require the presence of two different compatible cultivars for pollination to result in fruit production. Pollen can be transferred by wind, insects, or birds. Many fruit trees are pollinated by insects. Nut trees are usually pollinated by wind. After pollination, the pollen grain must germinate to lead to fertilization. The success of pollination and fertilization depends on favorable environmental conditions. It also requires the right pollen grain reaching the right flower, as pollen can only fertilize specific compatible flowers. For example, pollen from a peach flower will not pollinate an apple flower. Cross-pollinate Cross-pollinating fruit trees should be planted no more than 60 meters apart. This will ensure that bees visit the trees often enough to ensure pollination. Nut trees need to be planted no more than 15 meters apart. A further complication is that a few apple trees and pear cultivars known as triploids produce sterile pollen. Triploid cultivars have three sets of chromosomes within their cells. A triploid cultivar will require another cultivar for pollination, and the trees must flower at the same time. So in part A, they have asked, explain the difference between self-pollination and cross-pollination. So this is a very straightforward question. Just we have to state out the difference. That is, self-pollination occurs within the same plant and cross-pollination occurs between different plants. Then we have describe the sequence of events from pollen landing on a flower to fruit production. So first, when the pollen lands on the stigma, it releases digestive enzyme which digests the style to produce a pollen tube which grows down the style and joins with the ovule through a gap which is called micropyle. So this allows the pollen nucleus to go down the style through the micropyle and reach the egg to fertilize it or fuse with it so it can form a fruit. And the rest of the part becomes flower, or sorry, seed or something else. So here, pollen tube grows down the style, then it enters the ovule through the micropyle, allowing the male nucleus to travel down and fuse with the male gamete. Ovule becomes the seed and ovary becomes the fruit. On to the next question. Fruit trees often produce sweet tasting fruit containing sugars. Sugars are carbohydrate. Name three elements of carbohydrate, which are carbon, hydrogen, and oxygen. We have to just write out those three. Then they've asked, suggest how production of sweet tasting fruit may help spread the seeds of a fruit tree. So if a fruit is sweet, then more animals will come and eat them. Then the seeds they have eaten will be not digested because most of the animals do not have the capability to digest cellulose or the seed. So they will ingest it and the seeds will be dispersed around a bigger area so they can grow into a new plant. So fruit trees are eaten, but fruits are eaten by animals, then ingested, and the seeds get dispersed. Explain how self-incompatibility prevents self-fertilization. So we know, as we have read in the passage from line 11 to 13, that they cannot fertilize themselves. So it happens due to the pollen, which cannot grow a pollen tube. So the male nucleus cannot go down and fuse with the ovary. Thus, no fertilization occurs. So pollen of the same plant cannot grow the pollen tube, so no fertilization. Then describe how the structure of insect pollinated flowers help to achieve pollination. So we know insect pollinated flowers have large, colorful, and scented petals to attract insects, and they may produce nectar to also further attract the insects. So this is the answer, large petals which are colored and scented and produces nectar. Give a reason why nut trees need to be planted closer together than fruit trees. So we read in passage line 23 to 25 that nut trees are pollinated by wind. So they're wind pollinated flowers. And at the last part of the passage, we read that wind pollinated flowers need to be planted no more than about 15 meters apart, and cross pollinating flowers or insect pollinating flowers should be no more than about 60 meters apart. So that should be the answer why they are closer than the fruit trees, because they are wind pollinated flowers. And the last question of question number 1, explain why triploid cultivars produce sterile pollen. So we read about triploid cultivars that they have 3 chromosomes. So if they wanted to produce a sterile pollen, they had to undergo meiosis, because pollen is a male gamete and male gamete is produced by meiosis. But triploid cultivars have 3 chromosomes. 3 cannot be divided into half or made into half, so they cannot undergo meiosis. Thus, the male gamete of triploid cultivars cannot be produced. So they cannot divide by meiosis, so they cannot produce the haploid gametes. So this concludes question number 1. Let's move on. In question number 2, we have a diagram of an eye. Name parts W, X, and Y. W is the lens, X is the cornea, Y is the retina. So that is our answer, lens, cornea, and retina. Describe how structure Z, which is iris, changes when a person moves into a dark room. So how it changes, there are 2 muscles present. One is circular muscle, sorry, and other one is radial muscle. So when a person moves in the dark room, the circular muscle relaxes and the radial muscle contracts so that the pupil dilates. So more light can enter their eye to enable them to see in the dark. That is our answer. Circular muscle relaxes and radial muscle contracts to dilate the pupil. Then on to question number 3, we have a student uses an apparatus to investigate the water loss from a plant. So this is a weight photometer. So here, let's see the steps they've used to measure the total mass of the flask, water, oil, coin, and cotton wool and plant. Leave the apparatus for 48 hours to measure the new mass and calculate the rate change of mass. So repeat it for 4 times. Name the piece of apparatus used in this investigation. As I've said, it is a weight photometer. So then they've asked us to the a math calculate the gram per hour rate change of mass. So the total mass changes from this much to this much. They've given us the change in mass and the hour and the time. So for rate, we have to divide the change in mass divided by time. So we have to find the difference between these two and divide by 48 to get our answer, which should be to two significant figures. So this is the answer, 0.057. Explain why student repeats the investigation with the same conditions. We repeat any experiment to increase the reliability by allowing or by calculating the mean so that we can avoid anomalous or inaccurate results. So calculate mean, and to increase the reliability. The student changes the conditions surrounding the plant. Describe how the student could investigate the effect that moving air has on the mass, change in mass. So if the air is moved, sorry, we want to investigate the effect of moving in the change, the effect of moving air on the change of mass. And the student, and they've asked us to describe how the student could do this. So we could use a fan to move the air. Meanwhile, we should also keep the other conditions same, because if we do not keep the other conditions same, then the other factors may affect the change in mass. So we only want to figure out the effect of moving air on the change in mass. So the answer is use a fan while keeping temperatures and other factors same. Explain the effect that faster moving air has on the change in mass. So if the air is moving faster around the plant, that means the humidity around the plant will be lowered, which will increase the rate of transpiration, thus decreasing or changing the mass much faster. So more mass will be lost. So this is our answer. More mass is lost due to increased transpiration as humidity decreases. Describe how the student could modify the investigation to study the role of roots in the loss of water from a plant. So now we want to study the role of roots on water loss from a plant. So first we should take, to modify the investigation, we should take two plants of the same species and remove the roots to investigate or analyze how much water they lose from a plant, or we can analyze the role of them on the rate of transpiration. So this should be our answer. We just have to say what we have to do. We will take two species of plant and remove their roots for 2 marks. Then we have question number 4. A group of students investigate the biodiversity in two fields, Field A and Field B. Field A is grazed by cattle and Field B has no cattle. So grazing means cattle eating the grass or plant or everything they ate, whatever the cattle eats. So grazing means being eaten by cattle. First, they've said us explain what is meant by the term biodiversity. So we know biodiversity is a measure of variation in organisms, or we can say the number of different species and the number of each species in an area. So for 2 marks, let's go for that. Number of different species and number of each species. In part B, the student investigates the plant species in each field. They've counted the number of each species in 0.5 meter into 0.5 meter quadrate. So they took a quadrate and used it, repeated for 5 times, and investigated the number of each species present in each field. So this is the result. This is field A, repeated at five times, and these are the species and number of species that are present in both fields. So let's analyze it quickly. For field A, most of the species are present except common sorrel and stinging needle. And in field B, most of them are absent except meadow buttercup and common sorrel, white clover, and stinging needle. So we can see field A has more number of species, so it must have a higher biodiversity than field B. So let's see what they've asked us. Describe how quadrate should be used or placed to obtain unbiased species number for each field. So we have to use quadrate by placing it at random coordinates so we receive the unbiased result. So we do not have any favorite spot to place it in. So we will use random coordinates or place it at random coordinates to get the unbiased result. For most frequently occurring plant species in field B, calculate the number of plants in 1 square meter. So we have to find the most frequently occurring species and that too in 1 square meter area. So let's see the table. What is the most frequently occurring species in field B? That should be common sorrel because they have the highest number, about 25, 25 plus 10, 35 of them in about 1, 2, 3, 4, 5 into 0.5 into 0.5. That is the area of the quadrant. So let's break it down. To calculate the number of species in 1 meter square of area, we have to find out that at how many areas or at what area were these 35 species at. So first, each quadrant is about 0.25 meters square, the area of each quadrant. How did I find that? We know they've told us that a quadrant is side by side 0.5 into 0.5. So area of one quadrant should be 0.5 into 0.5, which is 0.25 meters square. So this is repeated for 5 times, or we can say all these 35 species have 35 common sorrel have been found at 5 different areas. So the total area should be 5 into 0.25 or 0.25, which gives us the total area where all 35 species, all 35 number of common sorrels were found. So now we know that at 5 into 0.25 meters square, there are 35 common sorrels. So how much would there be at 1 centimeter 1 meter square of area. Then we should divide, to find that we should divide the total number of common soils by the total area. That way we will get the number of uh, plant or common soil in 1 meter square. So that's what we did here. The total number of common soils divided by the total area to give us how many common soils or how many number of plants were there in 1 meter square of area. Next question we have, name the species with greatest variation of plants recorded in quadrant for field B. So in field B, we can see there are none. There is some variation, but the greatest variation should be for meadow buttercup. So the answer is meadow buttercup. The student could conclude that grazing affects the species in field A. Using the information and your own knowledge, discuss the student's conclusion. So we have seen in the table A or field A, they had greater number of species, greater number of each species. You can see there are many number of species present except common sorrel and singing needle, but they had more number of species than field B and more number of each species than field B. Therefore, we can say A has greater biodiversity. And since we've read that field A was grazed by cattle, we can say then grazing by cattle does increase the biodiversity. But we still do not have any information about the cattle or what cattle were they cows or goats or sheeps. We do not know any information. And further, it was repeated about 5 times, so we can say it is somewhat reliable. So our answer for 5 marks will be like this. In field A, there were more species present, more even distribution, although there were some of them absent. But overall, they have the greater biodiversity, so we can say grazing does increase biodiversity. And it was repeated about 5 times, so it is reliable. But further, we do not have any information about the animals or the cattle or any types of fertilizers, water, or how much hours of sunlight was available at each field. We do not know that. So this is the answer for our last question of question number 4. Question number 5. They gave us some data collected to measure how successful cloning in cattle was. So there were about 3,374 embryos transferred, and only about 317 of them lived, or live calves were born. And then those live calves survived about 279 after 24 hours, and after 150 days, about 225 of them were alive. Calculate the percentage of embryos transferred that resulted in live calves. So they're asking us how much percentage of the embryos survived or were transformed into live calves. So to find that percentage, we have to divide 317 by 3,374 and multiply it by 100 to find how much of those embryos survived. So this is what we will do. We will get 9.4%. In the next question, they've asked us to describe the stages in the production of a cloned mammal from an adult mammal. So we know to clone a mammal, we have to take the nucleus of an adult cell and transfer it to an enucleated egg cell, join them by electric shock, so that they can divide by mitosis and form an embryo, which will be then transferred to the uterus of the surrogate mother. Then they've asked us, there is some evidence that cloned cattle may have weakened immune system. Explain why a weakened immune system can affect the health of a cloned cattle. So, how can a weakened immune system affect the cloned cattle? Weakened immune system means they must have lower number of white blood cells, which will result in lower number of antibodies, so they cannot fight infection or hold up against infection much or very in a proper way. So they will be catching diseases more often. So there are fewer red blood cells and fewer antibodies produced. Scientists also collected data comparing cloned cattle and control group cattle. So we can see the difference between two of them, that uh, there were about 10 cloned cows and 11 control number of cows, which is small but about similar, but the mean birth mass was greater for cloned cows. Mean age at puberty in days, that means the age, or how many days later did they hit puberty. This is for cloned cows is 419 days and 356 days. So cloned cows hit puberty later, and mean body mass at puberty. So mean body mass is greater for clones, and daily increase in mass is about the same for both clone and control group. Comment on the effect of cloning on growth and development of this cattle. So for growth and development, we can see that our cloned cows have greater mass birth and reaches puberty later than control groups, but has mean body mass, greater mean body mass than control groups. So they're about greater in these aspects, but as they've said us to comment on the effect, we can say this effect might not be, or these conclusions may not be much more reliable because it was not repeated. There is no sign of repeat, and this is a very small study because only 10 or 11 cows were used, very small number. So this should be our comment. Small study as few cows are involved, not repeated, so may not be reliable. But as the data or the table shows, higher birth mass in clones, later puberty in clones, and higher mass at puberty for clones. So that concludes our question number 5. Let's move on to our last question, question number 6. This is about nephrons. The diagram shows a nephron from a human kidney with some structured label. Which of this is structure U? Structure U should be proximal convoluted tubule. So this is answer D, or option D. Which process occurs between S and T? S and T, so this is glomerulus and Bowman's capsule. So the process that occurs here is selective reabsorption. Sorry, no. This is the process that happens here is ultrafiltration, because the proteins or large molecules get filtered here, so they cannot enter the nephron. So our answer is T. Structure where Which structure is the collecting duct? Collecting duct is this part, which is our label structure Q. So Q is option B. Then they've given us a table showing the number of the concentration of substance in gram per 100 centimeter cube. So in blood plasma, this is the amount of protein present in blood plasma, glomerular filtrate, and urine. And for glucose, they've given us the concentration also. Let's see what they've asked. Just calculate the mass of glucose in 5 dm cube of blood plasma. So in 1 dm cube, there is about 10, 1000 centimeter cube. So, and in our blood plasma, there is about 0.1 gram of glucose per 100 centimeter cube. So to find out how many grams of glucose there is in about 5,000 centimeter cube, we have to divide it by 100 centimeter cube to find out how much there is in glucose. So our layout should be like this. There is about 0.5 is 0.1 glucose in 5,000, which is 5 into 1,000. That is, we are converting the dm cube into centimeter cube. 5 into 1,000 divided by 100, because there is about 1 gram or 0.1 gram in 100. So in 5,000 centimeter cube, we have to divide it by 100. That gives us 5 grams of glucose. Explain why there is no protein in the urine. Because, as I've said, in ultrafiltration, which occurs in glomerulus, the proteins are big molecules, they get filtered out. So for 2 marks, we will write there proteins are large molecules, so cannot pass out of glomerulus. And the last question, explain why there is glucose in glomerular filtrate but no glucose in urine. So first of all, as glucose is present in glomerular filtrate, that means it can pass through glomerulus. That means they are small molecules. So glucose can pass through the glomerulus, but they are not present in urine. This is because they are selectively reabsorbed into the blood by active transport in the proximal convoluted tubule. So that's why they're not present in the urine. So glucose passes out of the glomerulus because they're small, obviously, then they're reabsorbed back into the blood by active transport, which is why they're not present in the urine. So this concludes the paper. I hope you understood everything I've said. Hope to see you on the next paper. Goodbye.